Thank you very much, everybody, and thanks to the organizers for inviting me to come and talk to you today about the genetics of EB. It's especially pertinent today because GeneDx has recently changed their entire platform for EB testing, so I'm happy to talk to you a little bit about that today and the kind of results that we're getting. But first, let me say that I've worked with GeneDx now for 9 years, that it is a publicly traded company owned by BioReference Labs, and we provide testing for a large number of rare disorders, including EB. And GeneDx recruited me when I left Jefferson to be their in-house expert on EB, and I've been really, really blessed to be able to do that from my home for the last 9 years. Okay, so DNA. I know this is like a foreign thing to most people, and you're going to have to bear with me as I learn how to use this thing here. So what is DNA? DNA is actually this sort of ropey, twisty thing. And the business part of DNA, let me see if I can go back, is the part in the center. Right in the center. And the center part is where the two sides unzip, and the center part is what codes. That's the pointer. Oh, good. Oh, yay. Okay. So the center, that's much better. The center part is the part that is the coding part. And the ropey parts on the outside are just the backbone that holds it together. And when DNA replicates, it unzips down the center. And the part in the center codes for the new strand. Okay? And it looks like this in 3 dimensions and with motion. And I just like to do this because I think it's a really cool slide. Okay, so genetic testing. When I started doing genetic testing, I started at the University of Pennsylvania in 1987, and genetic testing was in its infancy. We worked with single nucleotide or single base variants, and we were very, very limited in what we could do as far as genetic testing. We started with sickle cell anemia, Duchenne muscular dystrophy, and cystic fibrosis, which are the most common, among the most common genetic disorders. And then as time went on, we moved to full sequencing of an individual gene. And we've been doing it that way for a large number of years. From when I started at Jefferson in 1995 to almost the present day. We still do single gene testing in some cases. Then testing moved to a targeted gene panel, which is true of EB, where we look sequentially at the genes involved in EB. And then we moved to whole exome sequencing and what I'm going to discuss today, which is whole exome sequencing slice. And so you can see we've evolved considerably. And today I'm here to tell you that things are much better. They're faster. And we can see a lot more sequence data in one step. So you're all familiar with this picture. EB is a blistering disorder. It's inherited. And its estimated incidence is between 1 in 10,000 and 1 in 20,000, but that's a very soft number. It's being increasingly reported by physicians. There's no ethnic or racial specificity. And the inheritance pattern can be dominant or recessive, and I'm going to talk to you about that in a minute so you don't get hung up in words that you don't understand. And the clinical severity is extremely variable. There are a number of manifestations of EB, as most of you know. Besides the skin, there are findings in the eye, including corneal erosions. There's dental anomalies. There can be respiratory issues with the tracheal epithelium. There can be gastrointestinal issues. In one form of EB, there can be muscular dystrophy, and in another form, there can be urinary tract involvement. Okay, so why do we do genetic testing? If you have EB, you probably, A, want to know what form of EB you have, and a lot of times you want to know, you want to classify it as to the severity, so you can find out what you can expect over time. If you do genetic testing, the mutations that we find can help you prognosticate how severe it is and how severe it will be in the future in some cases. It also allows us to determine what the inheritance pattern in your family will be, whether your children or your children-to-be are at risk, whether your siblings have a carrier risk, and it can help you predict what your recurrence risk would be if you have a child. It's also increasingly being used for the basis of gene therapy, and you'll be hearing more about that, I believe, in this session. Okay, so the most common scenarios that present for genetic testing, and this is the most common scenario, in which you have two parents who have a child who unexpectedly has EB, and they did not previously know that they were carriers for EB because there's never been EB in their family. And so this would be called recessive EB, and it means that each parent carries a mutation on a gene, and that this individual inherited one mutation from each parent, because you get two of each chromosomes from your parents. And so one of the two chromosomes carrying EB from the father and one from the mother goes to the affected child. And they're interested in finding out what is the risk to this affected child. And the risk in this case will be 25% of that individual being affected with EB. And it will be 50% of that individual being a carrier of EB, and 25% risk of it being neither a carrier nor affected with EB. And this is the most common scenario that we find, but we also find some other scenarios. In this scenario, one of the parents has what we would call a dominant form of EB, and the other parent has no history of EB, and they want to know what the risk is to their child. And because dominant EB is expressed in one mutation is expressed in every individual, the risk of this child receiving the mutation from the parent is 1/2, because this parent has one normal chromosome and one mutation-bearing chromosome. And the child will therefore has a 1 in 2 risk of getting the mutated chromosome from this parent, and would only get normal chromosomes from this parent. In some cases, we don't know what the risk to this parent, that this parent has dominant dystrophic EB. The only way that you might know that, if there's no other people affected in the family, is by doing genetic testing. So if you do the genetic testing and you find out that this individual has a dominant form, now you know the risk to this child would be 1/2 rather than 1/4. Okay, so it's very important. And you cannot tell just by looking at severity of the disease whether it's a dominant or a recessive form, nor can you detect whether it's dominant or recessive from a skin biopsy. The only way you can detect whether it's dominant or recessive is by doing the genetic testing. And in some cases, we have a scenario like this, where we know these two parents are carriers of recessive disease. They have one affected child. The sibling of that child has a partner, and they're going to have a child, and they want to know what their risk of EB is. And so the scenario here is to test this individual, find them two mutations, in this case it's recessive, find the two mutations, see if this individual carries one of the mutation. If this individual carries a mutation, then we can also test the unaffected partner and see whether that individual carries a mutation in the same gene. And then that occurs very rarely, because the carrier risk for dystrophic and junctional EBs are about 1 in 350 in the general population. But assuming that that individual is a carrier, then we can offer them prenatal testing. So these are some of the most common scenarios, but there are other, obviously, family constellations that we encounter. Now, this is just a really general slide. What I just wanted to point out to you, and I hope Dr. Lucky actually went over this, is that there are 4 major and numerous variant subtypes of EB. There's a new classification system out this year, authored by Dr. Joe David Fine, which goes into that in detail. But the important thing to note is that in EBS, there's a suprabasal and a basal form. The suprabasal form has recently been added to the classification, and there are a number of new genes that we now look at when we're looking at EBS, including transglutaminase 5, plakophilin, desmoplakin, plakoglobin, in addition to, in the basal form, keratin 5 and 14, plectin, exophilin, and BPAG1. Not so long ago, about 2-3 years ago, we only looked at the 10 major genes, and now there are 26 genes in our gene panel that we look at when we do EB testing. Okay, and then in junctional EB, there's 2 various forms, and there are 4 proteins involved, the laminins, collagen 7, and in the localized form, rarely you can see alpha 6 beta 4 integrin mutations. And in dystrophic EB, you see only collagen 7 mutations. So what I just want to say, and this is a very lot of information here, but there's all different kinds of mutations that we can find in most of these genes. And in some of them where there's a limited number of mutations specified, it's because we just haven't seen that many cases with mutations in that gene. But what this means is that there are nonsense mutations, which means we get a stop codon, which some of you will be familiar with that terminology, but I'll discuss it in a minute. There can be deletions. There be inserts and deletions at the same time. There can be insertions and what we call splicing mutations, which I'll also show you. And this slide is just to show you that we're looking at all different kinds of things. When we screen a gene or we screen a panel for mutations, we're not looking for just one thing. That's the kind of testing that was done back in the 80s, targeted mutation testing, where you knew a certain mutation caused disease in a certain proportion of patients. That's not what we do. We screen the entire gene and some of the surrounding sequence to see what we can find. And once we find something, we then have to decide whether it causes disease. And as you can see in the other forms of EB, there's also a large number of different kinds of mutations that we identify. And more, okay? And Kindler syndrome is a new disease that's been added to the spectrum of EB. And in fact, we have seen a couple of Kindler patients that were referred to us for EB testing. And these would have not been picked up by the previous panel that was done as recently as 3 years ago. Okay, so this is a picture of the skin, and what I want to show you here, and the message I want to relay, is that there are proteins that hold your skin layers together. And the proteins start, this is the dermis that's under the skin down here, and these are collagen 7 molecules that make up the anchoring fibrils. And they anchor the basement membrane, which is this structure here, down to the tissue underneath your skin. And mutations in collagen 7 give you dystrophic EB. In this layer, okay, which we call the basement membrane, the mutations, there are 3 major proteins that are found in this area, and they are the laminin B3, A3, and C2 proteins that are coded for by these genes. And they make up what are called the anchoring filaments, and that is attached down by the collagen 7. And then the next layer above, you find these structures called hemidesmosomes. And the hemidesmosomes have partially interior and partially exterior to the cell structures. And they are made up of a bunch of proteins that then anchor the cells themselves down to the basement membrane and to the dermis. So it's like a sandwich. You have a whole bunch of layers that hold your skin down and create a barrier between the dermis underneath and the environment above. And recently, the genes evolved in the suprabasal structures, including the desmosomes, which are found that hold the various cells in the upper layers of the skin together, are also known to cause a form of EB simplex. Okay, and this is just a fancier slide just showing you sort of how the proteins look, sort of what we think their structures are like. Collagen 7, laminin 5, BPAG2, which is collagen 17, and the intermediate filaments, which are keratins 5 and 14, which are inside of the cells to give them some toughness. And it just forms a network, okay? And if any one of these proteins is affected, such that they can't function properly, then you're going to get EB, okay? And any mutation that takes the protein completely away, okay, is going to be a more severe mutation than a mutation that allows the protein to be formed, but it just doesn't work as well. Okay, so that's what stop codons are about. Stop codons, when the protein is being formed, it stops protein synthesis prematurely, and then the protein goes out into the cell, usually, and is degraded, and never finds its place in the network, okay? And that means that there'll be a big gap, and that means your skin is going to be more fragile because the network is not there to hold the layers together properly. Okay, and part of what we do at GeneDx for finding mutations is also to provide the basis for prenatal testing for families who are at risk for EB. In some cases, they just want informed choices. In some cases, they want reduced anxiety as to whether the child is going to be affected or unaffected. Some families opt for C-section to reduce trauma to the skin, and it allows families at risk who wouldn't otherwise choose to start a pregnancy to understand that EB testing is available to them for a future pregnancy. Okay, until very recently, the first step of doing mutation testing for EB was to do work with a skin biopsy. And the reason for that was, as you will recall from the long list, there's a whole lot of proteins involved. And if we have to sequence those sequentially one at a time, not only would that cost a tremendous amount of money, it would cost a tremendous amount of time to do. So if you have a skin biopsy, in the past, you could find out which protein or which layer of the skin was affected, and you could narrow down the list. If it was dystrophic EB with a split where the protein is missing below the basement membrane, then you would look at collagen 7. If it was junctional EB, you would look at the laminin 332 genes or collagen 17, et cetera. So that was the recommended first step for genetic testing. Today, it's not necessary to do that. However, it is useful to us when we go to interpret the genetic testing results, and I'll tell you about that in a minute. So it's not that I don't recommend it. I do still recommend it. However, in many cases, it's not absolutely necessary. Okay, so exome sequencing. I don't imagine many of you have heard about that yet today or in this conference. But what that means is that we're sequencing the coding regions of the genes. We're not looking at regions of the genes that don't code for protein. And there's a whole thing called whole exome sequencing, which looks at, in one step, all 20,500 genes in the genome. And what we do and those genes represent only 1% to 2% of the genome, because there's a whole lot of sequence in your genome that is not involved with coding for proteins, and some of which we know what it does, but most of which we don't know what it does. In the past, we called it junk DNA. It is definitely not junk DNA, because evolution would not allow 98% of your genome to survive if it was junk. However, the genes, the coding genes in the exome, the expressed part of the genome, is only 1% to 2%. So whole exome sequencing which we call WES, is simultaneous sequencing of all of those genes. And this is what it looks like. So this is a gene down here, and the genes are made up of segments that code for protein called exons, and they are interrupted by segments that are called introns. So exomes sequencing would sequence just this, just this, just this, and it would not sequence the intron regions. Okay, and so when we do the sequencing, that means that things that are in introns and things that are outside of the express region of the genome, except for the immediate 1 or 2 bases on either end, will not be detected. That sounds bad, except for in 85% of genetic diseases are associated with changes in the protein coding regions and what we call the splice, acceptor, and donor sites, which are these regions on the very end, which code for bases that allow the machinery of the cell to hook the exons together and make a coding sequence, okay? So when the gene is expressed, these introns are removed, and the exons come together and make one big, long RNA, okay? So what we're looking at is the exons and the immediately flanking regions that code for the splicing machinery that allows them to hook together. So in whole exome sequencing, we use a sequencing method called next generation sequencing, and it uses a capture method, and I'm just, for those of you who care, we start with whole genomic DNA, we construct a library of that, okay? And we amplify it, and we immobilize it, and then we put it on a sequencer, which churns out the data. The old methods, which were highly labor-intensive and extremely time-consuming, are completely eliminated with this methodology. And what you get is, from a library of all of the genomic DNA in your cell, you get a sequence output, which is a raw sequence data. And the trick now is to take that and assign the sequence data to particular genes, to look for variants, and then to find out whether those variants actually cause disease. Because you have a whole lot of variants in your genome that don't do anything, or are very benign changes like hair color, eye color, the shape of your fingernails, all of those kinds of things. But what we're going to look for now is we're going to look at things that cause disease, and that's where it becomes tricky. Because we're going to see a whole lot of things, and some of them we're not going to be too sure what they are. Some of them we are going to be benign, and some of them we're not going to be sure. So the advantages of this is that we get a reduction in cost and turnaround time. We can do a whole lot of genes at one time. And almost all the genes in the genome are available for analysis depending upon your coverage of your library. And we can also, in some cases, see large deletions and duplications, which we could not necessarily see with the old methodology. So these are the limitations which don't really apply to EV, but there's classes of mutations that we cannot see, including mitochondrial genes and also some of the expansion repeat type mutations. But these don't apply to EB, and that's a good thing. Makes life easier for us. So at GeneDx, we get actually more people for whole exome sequencing than we do get for EB. And that's because children are born, they have a variety of anomalies that can't be assigned to any particular disease or disease spectrum, and they're trying to figure out what is causing the anomalies in that child. And so what they can do is they can sequence the whole genome, and they can look in that genome and see if they can find changes in genes that would address the anomalies in that child. Okay, so we do a whole lot of whole exome sequencing for diseases like that. Also, in some cases, there's not a genetic testing panel available for a particular spectrum, and we can do whole exome sequencing for that. However, in EB, we do a thing called slice. And what slice means is we take all of that exome data, and we just don't look at anything that isn't directly addressed by our gene panel. So we pluck out the sequences for the 26 genes that are implicated in EB. You can also, in some cases, if you have a child with anomalies and you want to develop a panel, this is not EB, you can also add other genes to the panel. Say if somebody has something associated with mental retardation, you might pick out certain genes for that. Okay, so whole exome sequencing is very flexible because you can pluck out the genes of interest, and the good thing for EB is you can look at all 26 genes in one step. So we have a whole lot of slices in-house that are available. The most common one that has been ordered at GeneDx is by far EB. And we started that testing about 2 years ago, roughly. So in exome slice, we find mutations in about 95% of EB cases. There were 7 cases out of the 124 that we've done where we found no mutations. 4 of them did not have the symptoms that were classical EB, and so a default to a whole exome test would be warranted. And 3 of them, as far as I know, because I don't always get good information, had EB symptoms, but we did not find a mutation. So that's pretty good, 3 out of 124 where we didn't find anything. The price, if any of you have dealt with this before, is $3,000 for all 26 genes. The old method of testing is $6,000 just for collagen 7, and between $1,500 and $6,000 for each of the other genes. So you can see this is a huge benefit in terms of the pricing and the coverage that we offer. If we do slice testing and we don't find the mutations, you can then order whole exome sequencing, which is $6,000, and you will get a $3,000 discount. So you will not have lost any money by ordering the slice first. Okay, so what are we looking for? Now we're looking for, as GeneDx, because our little mascot is the zebra, we're looking for the zebras in the herd. Okay, we're looking for the changes that cause disease or cause a phenotype in the herd. And these are the genes that we look at. These are the classical EB genes, and these are some of the new ones. And some of these haven't been associated with EB phenotypes quite yet, but we speculate that they will be in the future. So we look at a whole panel of 26 different genes. This slide is in here as a little commercial. And that is, if you give me clinical information, when I look at the sequence data, it makes it much easier for me to give an informative result. If I know this child looks like he has simplex or junctional or dystrophic, if he has pyloric atresia or not, if his skin is severe or not severe, if he's had a biopsy result, all of that information helps me look at the variants that I see, because in many cases I'm going to see a lot of them, and determine whether they're causing the disease or not causing the disease. So this is the workflow. So the sample arrives, we take the clinical information, the exome sequencing is done, and it goes to the bioinformatics team, and they basically do the alignments, pick the genes out that I'm interested in, and it gets uploaded to a server where I go in and I start looking at the changes that I see and assign them as to whether they're interesting or not interesting. Then I research them a little and decide whether they're disease-causing or not. Then we send it back to the lab, whatever we think is disease-causing, and they confirm it by the old method of sequencing, but just for a very small piece of DNA. And we do that because in the exome, we frequently get false positives, and we want to make sure that what we're reporting out is definitely a mutation. And although I've never had a false positive yet with exome, the West people have, so we always confirm it by Sanger. And then it goes into review, and I'm part of the review, and report writing, where I am the report writer. I interpret the data, and I sign the report, and then it gets sent to the physician. So that's the workflow. And usually this part of it doesn't take so long. Even when I review it, it doesn't take so long. But this part, because everything that gets done at GeneDx gets done in duplicate from here on. So we have several individuals looking at the data, confirming the data, looking at the literature, and then it comes to me for report writing, and then my report gets reviewed by the clinical director. Okay, so what kinds of information do we look at with exome sequencing? We look at the frequency of the variants in the population. We look at, there's a lot of information available publicly now due to the Genome Project. There's a thing called 1,000 genomes where they've sequenced actually many thousands of genomes. And if you have a variant and it has a relatively, like if it's found in 50 of 6,000 people in 1,000 genomes, we're going to know that in fact that is a polymorphism. It's not anything that's going to cause disease. But if it's not found in 1,000 genomes, then I'm going to look at it harder because that would indicate that it's something rare and it could be disease-causing. We can look at the mutations in this database called Human Gene Mutation Database, which is a curated database that looks at mutations in the literature. And we can look at the inheritance pattern, see if it fits with the phenotype, also the type of mutation it is. And then we also look at the sequence change with these computer-generated tools that tell us, is it likely to affect this particular protein? Is it in a region of the protein that is highly conserved? So what that means is if this protein is, part of the protein is conserved in humans, in chimpanzees, in rats, in zebrafish, et cetera, et cetera. If it's a highly conserved region of the protein, that means it's very important. If it's not a well-conserved region of the protein, in other words, there's variations all over as you go down through the phylogeny, then what you're going to see is what you're going to interpret it as not as important for the protein. Okay, so we look to see if it's conserved across species. We look at the gene function, biological pathways, and we make sure, and in our case, in the slides, that it's expressed in the skin. So this is what the one tab of the exome analyzer is. This is where we look at the previous medical history. This is the information that I look at before I do anything to see what the symptoms are in that individual, and it helps me get ready to look at the genetic testing data. And you can see we have primary indications, and in this case there was some prior testing done. So we'll be looking for that to see if we find those mutations as well. In this tab, it tells me that it has picked up results in each one of these categories. You can see there's 14 results in HGMD, et cetera, et cetera. So this is just a summary of what it's finding. And this is the actual data. So what you'll see here is a list of the genes in which variants have been found. You'll see whether this variant is heterozygous, in other words, only in one copy, or homozygous, or in this case, it's a duplication. And you'll see the mutation call. So what this means is, on this particular sequence, in this exon, this is the change right here, and this is a P11343L. So that is a missense change, and it's found in the collagen 17 gene. And so you can see in this individual there were a whole lot of changes that we identified. And the only things that stuck out to me, because I've looked at a lot of these, were this one, and CNV means copy number variant. So that means there could have been a duplication or a deletion in this particular gene. So the next, on the other side, that screen is long. So as you read across from collagen 17 here, it will tell us, automatically gives us the analysis data. Was it found in 1,000 genomes? Well, have we seen it in the exome analyzer before? How many homozygous individuals have been identified with that mutation? And then some of the tools that the computer generates to tell us whether that particular change causes disease. Anything in red means it's potentially bad, but what we're mostly looking at is things that are red all the way across, okay? Like here, okay? And that tells us we've got something that we really need to be looking at. And so this is what the data looks like to me. In the old days, anybody who's seen me giving a talk in the old days, I would throw up these slides with all these up and down squiggles that showed you sequence data. And I used to actually look through all of those squiggles and look through the calls for each one of those to find things in the mutation data. Now the computer does it all for me, because frankly, doing 26 genes like that would be mind-numbing. So the computer does all of that, and it generates this information for us. And then it gets summarized on this page, where it summarizes everything from that result page for a particular variant, and this is the variant here. And the review analysts will put in information, and I will put in information. And if the mutation has been seen before by GeneDx, we'll see that information up here, and it will help us figure out how to report that. And that report summary is there. So we can also detect copy number variants in this, which is areas where we have a low amount of sequence information. In this particular one, we actually saw this as no information on exons 4 and 5 of LAMB3. And sure enough, when we went back and did the confirmation, this individual had a homozygous, in other words, on both alleles, deletions of exon 4 and 5 in LAMB3, which would be expected to cause junctional EB. Okay, so what happens then? That information goes back for review, and then it comes back to me, and I report it out. In most cases, I will be able to say it is a pathogenic variant, and it is definitely associated with the phenotype. In some cases, we might see other mutations in other genes that could be associated with phenotypes, but that doesn't happen very often in EB. We might see variants in a candidate gene, or we might see other types of changes which might have clinical relevance but not EB. And this category is pretty much found in the whole exome sequencing rather than EB slice, because we're only looking at the skin genes, okay? So we wouldn't have a lot of other genes to look at. And so these are our results. In the past, you can see we did a lot of single gene testing here, and we started slice testing into the very end of 2012. In 2013, people started understanding what slice was about, and we started moving up. So the testing from this category up here started moving down into slice, and in 2014, we're getting the word out, and we now have 69 cases so far done by slice year to date, so I expect that number to be up over 150 by the end of the year. And we've done a total of 124 exome slice tests. Okay, so that's the end of my talk. I just want to say that I know some of you have particular mutations or genetic questions. I don't think we have time today to address them. However, I will be here and around the conference all through the day and the evening, and feel free to come up and interrupt me and ask your questions or get information on testing, and I'm happy to give you my email or my phone number, and you can even give it to your doctor. I'm readily available at any time. That's what I do for GeneDx to answer your questions.